Antibiotics have saved countless lives. Before them, even a minor injury like a cut finger could kill you due to a higher risk of sepsis. Childbirth was dangerous. Pneumonia was often fatal. But now there's a tension. Antibiotics can be life-saving, but they also disrupt your gut microbiome. Today, we're going to explore what antibiotics really are, how they work, what they do to your gut microbiome, whether it recovers, and what the actual evidence says. Stay tuned to find out more. Okay, so what are antibiotics? So here's a very quick history. Antibiotics didn't begin in pharmaceutical labs. They started in microbial ecosystems, especially in soils. Long before humans discovered penicillin, bacteria and fungi were producing antibiotic compounds, so millions of years ago. Now these molecules help microbes compete for space and nutrients in crowded environments. In soils, and even in the human gut, microbes constantly interact. So some release antimicrobial compounds to suppress competitors, so sometimes they use them as weapons and sometimes as subtle chemical signals. Now one famous example is penicillium fungi, which produce the antibiotic penicillin. Crucially, antibiotic producing microbes also carry what we call resistance genes. Now these are genetic tools that allow them to survive their own antibiotics, so their own weapons, by breaking them down, pumping them out, or altering their targets. So you don't want to shoot yourself in the foot. Now, when disease-causing bacteria acquire these same resistance genes, the antibiotics we rely on stop working. This is why antibiotic resistance or antimicrobial resistance is such a profound issue in today's world. So antibiotic resistance evolved over millions of years in natural microbial communities. What humans did was scale up production and dramatically intensify the pressures, so the ecological pressures that spread these resistance traits. See my book, Invisible Friends, for more on this. Now, in 1928, Alexander Fleming famously discovered penicillin after noticing mould killing bacteria in a petri dish. By the 1940s, antibiotics were mass-produced. They transformed medicine. They reduced deaths from pneumonia, they made surgery safer, they enabled things like chemotherapy, they reduced maternal mortality. They're considered to be one of the most important inventions in human history. What antibiotics actually are? Antibiotics are compounds that kill bacteria or stop them from growing. They do this by targeting specific bacterial features, such as cell wall synthesis. So penicillin does this. These antibiotics stop bacteria from building their protective outer wall, causing them to burst and die. There's also protein production. So tetracyclines, a form of antibiotics, these block the bacterial machinery that makes proteins, preventing the bacteria from growing and multiplying. There's also DNA replication. So fluoroquinolones, these interfere with the enzymes bacteria use to copy their DNA, so they can't reproduce anymore. And then there's folate metabolism. So trimethoprim disrupt chemical pathways that bacteria need to make essential building blocks for DNA, effectively starving them of what they need to grow. Now, human cells don't have bacterial cell walls or bacterial what we call ribosomes, so these are tiny protein factories, which is why many antibiotics can selectively target bacteria without directly damaging human cells. This is important, so antibiotics do not work on viruses. They do not treat colds, influenza, COVID-19, and many sore throats. Now, viruses are structurally and biologically different. They use your own cells to replicate. So, antibiotics have nothing to target. Now, using antibiotics for viral infections, A, doesn't help, B, promotes resistance, and C, disrupts your microbiome unnecessarily. Now, this is a major public health issue. This is just a quick interjection before we carry on. If you find this interesting or useful, I'd really appreciate it if you could subscribe to the channel. It really does help the channel grow and allows me to keep producing these educational videos. Okay, let's get back to it. Not all antibiotics are equal. We have narrow spectrum antibiotics that target specific bacteria. And then we have broad spectrum antibiotics. These target many groups at once. So broad spectrum antibiotics are more likely to cause widespread microbiome disruption in your body, but sometimes they're necessary in severe infections where the exact pathogen isn't known, for instance. So what happens to your gut microbiome? Well, your gut contains hundreds of bacterial species, trillions of cells. They break down fibre into short-chain fatty acids like butyrate, see my other videos, and they help train the immune system. And these microbes in your gut also contribute to colonization resistance. So some microbes can help block out pathogens from trying to colonize your body. They also produce vitamins like K and some B vitamins. When antibiotics are introduced reduced, several things can happen. 1, there might be a rapid loss of the diversity of your gut microbes. 2, there could be a collapse of susceptible species. And 3, expansion of resistant strains and genes. So remember I mentioned that antibiotic resistance or antimicrobial resistance is a huge public health issue. And 4, altered metabolic output. So when antibiotics reduce or reshuffle certain bacterial groups in your gut microbiome, the mix of chemicals that they would ordinarily produce changes, and this can affect things like digestion and other functions in your body. So let's look at some of the evidence. Study 1, deflacin. So researchers gave healthy adults a 5-day course of the antibiotic ciprofloxacin and tracked their gut microbiomes. Within days, microbial diversity dropped sharply. Some bacterial groups fell below detectable levels. After the antibiotics stopped, the microbiome began to move back towards its original state within weeks. But here's a crucial point. So recovery was often incomplete. When participants later took a second course, the microbiome shifted again and some changes persisted. Now, the community did not always return exactly where it started. So the take-home message is that the gut microbiome is resilient, but not infinitely so. So repeated disturbance can leave a significant mark. Study 2, Peleja et al. In this study, healthy adults took a 4-day cocktail of broad-spectrum antibiotics. The impact was quite dramatic, so microbial diversity plunged and some common bacterial groups were almost wiped out. Over the next 1 or 2 months, much of the microbiome recovered towards its baseline. However, at 6 months, several previously common species were still undetected in most of the participants. The take-home message here is that recovery can happen, but it may not be perfect. Some losses can be long-lasting. In an earlier study of clindamycin, researchers followed participants for up to 2 years after treatments. Although overall diversity improved, certain bacterial groups did not return to their original levels, even after long follow-up. Now, the ecological structure of the gut had shifted. Responses also varied considerably between individuals. The take-home message here is that antibiotic effects can persist far longer than symptoms, and recovery is highly individual. A note on colonisation resistance and Clostridioides difficile. One of the clearest real-world consequences, Clostridioides difficile infection. So antibiotics reduce diversity. Reduced diversity weakens colonisation resistance in your gut microbiome. This means that the disease-causing C. diff bacterium can then bloom. Fecal microbiota transplantation, or FMT, helps to restore diversity and is highly effective for recurrent C. diff infections. This demonstrates that microbiome structure has real clinical consequences. Does the microbiome fully recover? This is one of the most common questions. The evidence suggests that, in healthy adults, substantial recovery usually occurs, often within weeks to months. But composition may shift permanently. This can be really important for your health. Also, rare species may not return at all. In early life, the picture is more concerning. So, infancy is when the microbiome and immune system are co-developing. Several large human cohort studies have shown that antibiotics in early life can alter how the gut microbiome develops. In one study, published in Nature Communications, repeated macrolide antibiotics were linked to long-term changes in gut bacteria and higher rates of asthma and increased body mass index. Animal studies strengthen this case. So in a 2014 Cell paper, mice given early life antibiotics showed altered metabolism and gained more fat. In humans, early antibiotic exposure has been associated with asthma, allergies, and obesity. But it's important to be careful, so association is not the same as proof of causation. Antibiotics may be one piece of a much larger developmental puzzle, but the signal is consistent enough to warrant caution. Resistome expansion. Antibiotics increase antibiotic resistance genes in the gut microbiome. Now, some of these can persist long term. This is shown in multiple longitudinal studies, and it has broader implications beyond the individual. So remember I mentioned antimicrobial resistance is a huge public health issue, and I'll do another video on antimicrobial resistance soon so you can learn about this major public health concern. Why does the gut microbiome recover at least somewhat? So here's something remarkable. Despite being hit by antibiotics, sometimes dramatically, the gut microbiome often rebounds. But why? Well, it's because your gut is not just a bag of bacteria. It's an ecosystem, a dynamic ecosystem. And ecosystems have a level of inbuilt resilience. So in ecology, resilience means the ability of a system to absorb disturbance and reorganize without collapsing into something entirely different. Now, your gut contains dormant microbes that can sometimes survive the treatment, resistant strains that expand temporarily, microbes hiding in mucus layers and biofilms inside your gut, and species reintroduced through food and the environment. After antibiotics, what's left begins to regrow. So ecological niches open up and food becomes available for the microbes, and competition reshuffles. You can think of it as a bit like a bushfire. So some trees die, but some seeds survive underground, and some species return from surrounding areas. The landscape changes. It doesn't necessarily become a desert. Recovery depends on several factors though. So age is really important. You can recover, but it takes longer as you get older. Diet, especially fibre intake, which feeds those beneficial microbes in your gut. Environmental exposures as well are potentially really important, as are repeated antibiotic courses, and of course baseline diversity, so who's already living there in your gut in the first place plays a key role in whether your microbiome will bounce back. Now, diet plays a major role, as I mentioned, so fibre is not food for you, as I mentioned in several other of my videos, it's food for your microbes. When you eat diverse plant fibres, bacteria that produce short-chain fatty acids and other chemicals are more likely to recover. A 2021 study published in Cell found that eating fermented foods increased gut microbiome diversity over 10 weeks in healthy adults. Probiotics are more complicated. So in a 2018 Cell study, researchers found that after antibiotics, a standard commercial probiotic delayed the return of participants' original microbiome compared to simply waiting. The probiotic bacteria colonized temporarily, but native microbes came back more slowly. This doesn't mean that probiotics are harmful. It means the effects are strain specific and context dependent. The microbiome is more ecological than we often assume. So the idea that just take a probiotic and you're fine is overly simplistic. Now, recovery happens because the gut is a living dynamic system shaped by competition and cooperation and food supply and exposures. But resilience has limits, and these limits are being breached more and more in today's industrialized world, and repeated disturbance can gradually shift the baseline. So what should we do? Antibiotics are essential, but the lesson is not fear, it's precision. So use antibiotics when medically necessary. Avoid them for things like viral infections, and support recovery through fibre-rich diets, diverse plant intake. Biodiverse environmental exposures may also help, but more research is needed there. So getting out into nature, and avoid unnecessary repeated courses of antibiotics. And also recognise that we are ecological systems, so antibiotics are disturbances, and many ecosystems recover, but repeated disturbance can reshape them. 3 key takeaways from this video. 1. Antibiotics save lives, but they can be broad ecological disruptors. 2. Most adult microbiomes recover substantially, but not always completely, and early life exposure may have longer term consequences. And 3, the goal is stewardship, not necessarily avoidance, so use antibiotics wisely and support recovery with diet and environmental diversity and looking after your walking ecosystem. If you found this video interesting or useful, I'd really appreciate it if you could subscribe to the channel. It genuinely helps the channel grow and allows me to keep creating these evidence-based videos. So please click on the subscribe and also like if you liked it. Thanks. And check out the new Nature Gut Brain website at www.naturegutbrain.com, and feel free to sign up to my free newsletter at www.jakemrobinson.com. And if you like these interconnected concepts where we're talking about the symbiotic relationships between the visible world and the invisible world, then you'll probably like my books. They're up there. So books like Invisible Friends and Treewilding, and my newest one, The Nature of Pandemic. So please check them out too. And remember, stay wild inside and out.